All right. Yeah, first of all, I'd like to thank Dr. Gu for the invitation. It is a great pleasure to share some of our recent research in this AI foundation model and prompt-based learning for analysis of biomedical data. So I'm going to first of all introduce some basic concepts of foundation models and prompt-based learning. Then I will talk about biological knowledge mining using ChatGPT. And then I'll talk about this biomedical image analysis through prompting this foundation model called SAM. And then there are also many other foundation models. So I will talk about a few other models as example. Then I will summarize and provide some future work. So this foundation model, you probably heard a lot about them. For example, the ChatGPT is the one big example. There are many ways to describe AI and related models. Regarding today's topic, I like this classification of machine learning paradigms. The first generation of machine learning, according to this paradigm, is that so-called feature engineering. That is, you do this manual feature extraction. For example, for facial recognition, you see eye, nose, etc. And then you use some simple model like SVM, simple neural networks, to train a model. And then later is so-called architecture engineering. That's typical deep learning. In this case, you don't need to do feature engineering. You can provide raw data, either raw text, raw images, and feed them into deep learning model. And then you can get results accordingly. For example, this convolutional neural network, CNN, and LSTM are examples of this approach. Then third generation is objective engineering. So these are actually already large models, but people pre-train this model, and then they can fine-tune the model for a special application. For example, the image ResNet50 and the language model BERT use cases of such approach. And then the fourth generation is a prompt engineering. So like ChatGPT, the one example. So in this case, you do not need to fine-tune your model. You just prompt your foundation model and then apply it for your application through so-called prompts. Yeah, so here you see that the model looks like more and more complex. But in reality, through the prompt-based learning, it's actually easier for people to apply AI. So it's just like computer chips. So in early days, people have to use tubes, transistors, and then do a lot of kind of a composition engineering in order to use it. But nowadays, people just use like a big computer chips. It's actually a lot easier for people to use such technology. Same is true for these foundation models. Even though these models are massive, very complex, but the learning curve is actually a lot lower. So that's why it's actually very friendly for people to use it. Even people here, many of you are probably working in medical domain or biology domains, and then you can actually use such approach for your problem. There are many examples of foundation models. For example, the ChatGPT is a text-to-text. And then some people probably used this text-to-image method, like a DALL-E. That is, you can text something, and then it would generate image accordingly. And there are some other models, like this CLIP for connecting text and images, and the SAM for image segmentation, and the ESM for protein sequence. So the foundation model, of course, they are large models, but not all large models are foundation models. I believe there are at least 3 criteria for foundation models. First of all, it should be promptable. So prompt, just like ChatGPT, you ask questions, or in a more complex form, you can provide some input to your model. But a lot of large models are not friendly to be prompt. Second of all, foundation models need to be adaptable for many downstream tasks. So as an example shown here, that one foundation models trained on many data, can be used for all kinds of tasks. And then third of all, foundation models should have so-called emergent intelligence. That is, it can figure out something new. Just like students here, when you learn something, you first of all practice the homework, and then you can do similar problems. But then it requires many more practice or thinking in order for you to solve real-world problems. Those real-world problems could be quite different from your homework setting. Same is true here. By training on a huge amount of data, these foundation models often develop such an emergent intelligence so that they can solve quite different kind of tasks in real world that are not present in your training. So this problem-based learning, first of all, is actually use your foundation model as a black box. Sometimes people say black box is bad because you do not understand what's going on. But it actually substantially reduces the usability. For example, you have a phone, there are a lot of complexity in it, but you use it literally as a black box. You just interact with the phone without opening it or tuning it. So foundation model is the same way. That is, you do not have to worry about the details or tune it. You rather use it directly. Of course, you need to have a lot of interactions with the model in order for you to accomplish your tasks. In this case, there are different kinds of prompts. For example, you can have a so-called hard prompt. Like ChatGPT, your questions are hard prompt because you do not really tune your question. And soft prompt basically means that you have some vector or matrix. You train the prompts using some small data, and then you enter your prompt to the foundation model in order to get the results. So the prompt-based learning that you use these prompts to achieve the tasks. There are different kinds of techniques. For example, you can transform your input. You can provide some template, or you can add some layers on top of foundation models. So before this prompt-based approach, people often say, in order to do deep learning, you need three big things. You need a big data, you need a big model, and you need a big compute or computational resources. But with prompt-based learning, things are changing. First of all, you may not need big data. You can use a small amount of data to prompt your foundation model in order to do tasks. And then the model side, of course, you build on top of a big model. But then the additional, this setting is actually rather light. So it's a kind of mixed model. And then the computational resources are much less demanded comparing to previous models. So that's why this approach is very versatile to many applications and very friendly to academic labs or hospitals. And that's why I think even you are not expert in AI, you should be able to use these models for your own research problems. So let me move to this ChatGPT example for biomedical knowledge mining. So here I show this basic idea of ChatGPT. Since all of you already know a lot about it, I just want to say the basic idea. That is a ChatGPT model is first trained on free text. Of course, that's tons of text, like 45 terabytes of text. But then it applies this reinforcement learning from human feedback, or RLHF, for aligning the model. That is, you have some standard question and answers, and then ChatGPT utilizes standards to make the model like a more sociable. That is, you can interact with ChatGPT as if you are interacting with a human. And also through this process, it gains this reasoning capacity. This is so-called emergent intelligence. So typically, people use ChatGPT as a chatbot. Sometimes people use it for knowledge base. For example, if you have a new pet, you want to know how many meals a day you should feed your new pet. So these are actually knowledge base is more like a more sophisticated version of Google. So it's a search of something. But then from a research perspective, we actually focus more on the other aspects. One is a knowledge graph. That is, you have a big picture of many components, for example, genes, biological pathway, and then whether you can retrieve information from ChatGPT. And then another one is the inference engine. So ChatGPT itself becomes the inference engine. It can actually use it for training model or for a framework for a lot of other things, which I'm going to show some examples. So before I talk about the main research we are doing, first of all, I'd like to show some examples. People use ChatGPT for biomedical applications. First of all, education is obvious. For example, we do biomedical research. A lot of people in my lab are from computer science side. They need to know biology, and then ChatGPT is often the first stop in order to get biology knowledge. And then it also helps people in writing research papers. I think it's a more bigger version of Grammarly, so you can polish your paper a lot. So before that, even though my English is not that great, I spend tons of time revising student languages. But now I don't have to spend that much time in languages. I can focus more on the content. And then I have some couple of other examples. So both examples is actually in collaboration with Professor Gang-Jin Hu at the West Virginia University. So he actually wrote the first paper of applying ChatGPT to develop biomedical pipeline for data analysis. So I was invited to write a comment for his paper. And then after that, we and some others were interviewed by Nature. And Nature actually wrote this paper, 6 tips for better coding with ChatGPT based on our input. So if you're interested in it, you can take a look how ChatGPT can help you to be a better programmer. Another ongoing research we are doing that we actually systematically study programming based on ChatGPT and to see how often they can be reproduced, how often they can be successful. So eventually we want to build automatic pipeline so that people can pretty much talk about what they want to do, and then ChatGPT can build entire biomedical pipeline for you. So we're not talking about just 1 or 2 steps. So in this case, we have like 10 steps, like ChIP-seq, RNA-seq, etc. It handles them all. Another work I did with Dr. Hu is called a panel GPT. So this is a chatbot application in sports rehabilitation. So in this case, athlete may get injured, so he or she needs to know what to do. Or in some other cases, it could be in a classroom setting. People want to learn how to handle this situation. So we actually simulate a panel discussion using ChatGPT. So the ChatGPT has basically 5 channels. One is a therapist, one is a psychologist, and then nutritionist, AI expert, and athlete. Either the athlete or the discussion leader can propose a question, and then all these 5 channels can discuss, and later it automatically summarizes. It turns out it really can assist healthcare professionals with a lot of useful information. And this paper actually is already accepted and will be in the JMRI very soon. So here is actually what we do with ChatGPT a lot. That is mining ChatGPT and the knowledge graph. So in today's world, we have tons of literature information. Some of them actually are in the graph representation, like a picture. I'm going to talk about how to mine graph through image understanding later. But then a lot of it is in text. So traditionally, there are rich research in mining the text to find gene relationships, gene disease association. So in this case, we want to see whether we can treat ChatGPT as a knowledge graph, and then we'll mine them directly for knowledge discovery. For example, for kinase substrate relationship, etc. We believe because ChatGPT was trained on a lot of medical literature data, for example, archive or bioarchive, and also the PMC is open source, open access literature. So it should contain a lot of information. And then the challenge that we all know ChatGPT has this hallucination issue, that is sometimes give a completely wrong answer or fake answer. So we want to do a systematic research on this. Here we want to propose a question, that is what effect does gene A have on gene B? And then we design prompts to get the best results. In this case, we do have ground truth. It's a CAC data. It's a biological pathway database. And then we choose 200 pairs for training, 50 for validation, 50 for testing. This actually is not for the purpose of retrieving the existing information, because the information is already in the CAC database. Our purpose is, first of all, to evaluate ChatGPT for knowledge discovery. Second of all, by using this, we could discover new relationships. And we do not do it through this web interface, but rather we do it through API. That is, we query through command line by using the GPT 3.5 Turbo. And then if we can ask questions directly, we probably still need to restrict the answer a little bit, because without restricting it, it will give you lots of materials. So here we just want ChatGPT to answer in a very definitive way. Basically, we provide 4 options: gene activation, gene inhibition, phosphorylation, and no information if ChatGPT doesn't have a basis to make a prediction. So there are only 4 cases. Then we ask ChatGPT not to explain, just give the answer. So we actually designed some prompts for this. Nowadays, the prompt-based engineers actually became a highly paid profession, because it's actually very sophisticated. There are dozens of prompts, methods to define better prompts. For example, here, we use some of them, like the role prompting. You just say, as a computational biologist, determine what is the relationship between gene A and gene B in CAC pathway database. And then there are other methods like a generated knowledge prompting. So here you can either use some existing knowledge, or you can actually use the output of a previous session of ChatGPT as input here. And then you can define a certain format, certain direction. So in this case, gene A activates gene B is not the same as gene B activates gene A. So we define the direction. And then another important prompt is the so-called few-shot prompting. Here we give some examples. What effect does gene EGF has on gene EGFR? So this is activation. So we define these 3 examples. Of course, for machine learning, such data is not sufficient to train, because you only have 1 example. But it really defines the format and the workflow. And then now you can ask questions. Of course, you can provide more text to ChatGPT. For example, phosphorylation, and this is a biological term. You could actually provide a lot of information so that ChatGPT can understand what it is a little bit better. Then let's see the results. First of all, we found ChatGPT is actually very sensitive to the format. For example, you have this queries, and then in the few shot, you have this format Q and A, like the question and answer. If you ask question according to the format, it actually gives you a very good result. If you don't follow the format, it gives you a very bad result. So the prompt 1, it doesn't follow this format. And then it it actually 98% of chances it gives you a result of no information. So ChatGPT, if you ask multiple times, it gives you sometimes different answers. So here is we ask ChatGPT 100 times. Almost all cases, it says no information. But if you follow this format, 95 times gives the correct answer. So it's very sensitive to the format. And another thing that you actually use different prompts, you get quite different results. In fact, with the null prompt, that is, you don't really give many tricks. It still works reasonably well. So you provide more tricks like this prompt I talked about, few shots prompts, etc. It's actually get a lot better result. So this F1 score is actually a balanced measure about the overall performance. So you do see some improvement. And then you provide a lot of explanation, as I said, it's a phosphorylation. You found that the F1 score overall is actually became worse, but the precision became better. So what precision is that you predict a few of them, and then they tend to be correct. But a lot of the things you miss, so that's called precision. In this case, it actually reduced this hallucination, but it covers less results. So we want to actually optimize the prompt through this iterative prompt engineering. This is actually a novel approach nobody used before. So the idea that now we have this 200 training data, and then we can use the training data and then run ChatGPT 3.5 and get a precision recall F1, some false examples for each prompt. And then we can change prompt. Then we can feed this prompt into GPT-4, and then let the GPT-4 to figure out what would be a better prompt. So for example, give 5 prompts. Prompt 1, here is the prompt, here's the result. Prompt 2, here's the prompt, here's the result. And ChatGPT-4 can figure out. The reason why we use a different GPT model is that here we need to run many times. So we use a cheaper model. This actually only costs like $2 for 1 million prompts. GPT-4 is a lot more expensive. It's almost 20 times more expensive. So that's why we don't run this many times. And then we can actually define this format. Let's say we have this parameter called temperature. So when you have higher temperature for ChatGPT, it tends to generate more diverse results. The lower temperature, it gives you more concentrated result. So here, when we want to optimize the prompt, when we use GPT-4, we actually gradually increase the temperature, so it can sample more and more possible results. So this is a novel approach. You pretty much treat hard prompt like a soft prompt. We know that the hard prompt, like sentences, usually people do not tune them. But here, we actually let GPT-4 to tune those prompts like soft prompt. And we do it in a very rigorous fashion. So in machine learning, we typically have the training data, validation data, and the test data. So training data are those 200 pairs in our case. So each time we run 200 and get a precision recall F1 score. And then we generate this. And then we see that when we do this iterative prompt, it actually fluctuates, but it generates really good F1 score. So we validate this using the validation data, that is independent data. And also it shows the same result. So here, that means that this iteration achieves better result. So based on validating result, we get the prompt for this iteration. And then we use this prompt to run on a test data. So test data is like independent data. We only run it once and get the results. Indeed, through the refined prompt, it actually improved all aspects of this criterion. So it's a really great way to get a good prompt through such an iteration. And then we want to see what really changed by ChatGPT 4. So we look at the one of the best prompts. Here you see that previously we say as a computational biologist. Here it changed as a molecular biologist specializing in gene interaction. So it becomes more specific. And here you see this previously just say activation, inhibition, etc. It actually gives the bracket, says gene 1 activates gene 2, gene 1 inhibits gene 2, etc. So for us, this is just a redundant language. But for ChatGPT, somehow it's actually very useful. It really improved the performance a lot. Of course, ChatGPT, we found that the performance is not very stable because they keep improving their performance. We found actually, in our case, this year, the performance actually became worse over the past few months. So if you look at overall F1 score, in no prompt or manually designed prompt, it became worse. This actually is nothing new because other people found the same thing. That is the July version of ChatGPT has a lot worse inference capacity than March version. But if you look at it, the reason that OpenAI and Microsoft, they want to optimize the other side, that is to reduce hallucination. So here, if you use ChatGPT now, you found a lot of links, a lot of publication sites, they are real. While in early version, a lot of publications are completely fake. All the URLs are not real URLs. So it is actually a balance between reducing hallucination and improving inference engine. That actually remind us we probably should use a different language model for our purpose, which I'm going to talk about later. And these prompts actually not only can define two genes, their relationships, you can also get a chain of relationships through this so-called chain of thoughts method. This is actually the well-known prompt skill. And then you can get them all. So in our case, we can map the entire pathway. Of course, right now, we still cannot recover 100%. In fact, judging at whole pathway level, we can only cover 27%. But this is still a lot better than earlier text mining methods. So the point is that through ChatGPT, you can really infer the entire pathway or the entire biological mechanism, even though this is just a prediction, but it still provides a good hypothesis. So hallucination, from computational term, we call the false positives. Any computational predictions, you would have false positives. So this is nothing new. As long as the model, the prediction results give you useful hypotheses, they should be used. Next, I'm going to talk about this image analysis by prompting SAM. So SAM is another foundation model for image. Here, it is trained on 11 million images. The idea that you have this image and then you feed into this encoder. Basically, you transform the image into some kind of vector. The platform uses so-called vision transformer. And then you can decode this vector. For example, you can do image segmentation. And also you can segment at a different level. You can whole level of Caesar or part of Caesar, etc. So we actually utilized the SAM once it was released a few months ago. We actually applied it to image understanding. For example, here it is a figure in the literature. We want to use model to understand what's going on in the figure. For example, in this case, ATM activates KU80. But can we get such information from the figure automatically. To our pleasant surprise, SAM can do this extremely well. For example, here is the biological pathway, and SAM can do segmentation of all those components very well. Even like this, we had a hard time to get such a relationship. This actually is a we call T-bar. It represent inhibition, but it cross another segment, so it's not easy to get it through image segmentation. But Sam did a great job and then retrieve it. And then we use this approach to, first of all, segment the image, and then we get all these components. And then we use another large model, ResNet-34, to train the types of those images, whether it is a text box or whether it's an arrow indicate activation, or whether it's a T-bar, which indicate inhibition. So we automatically parse this. So we worked on this for several years, and the best model we could achieve previously was this RetinaNet. And you see that with SAM, all the measures got significant improvements. And many of the hard cases, SAM can get it right. So we also use it for other applications. So this actually is in collaboration with Dr. Jianling Chen's lab. So they developed this database for core-EM. So this is actually a core-EM micrograph for proteins. Each spot you see here represent one protein. And this actually, the ones in green shows their labels. That means the coordinate of the real protein. And in this case, they use a circle. It shows a diameter. So we actually use their data and get some training data and some testing data. And now question is whether we can do this automatically. Of course, the first one is that we want to apply SAM directly. So in this case, you put the image in, and then SAM can give you this segmentation. And then you compare with the ground truth manual annotation. In this case, it's so-called dice score. That is that you have a circle here, and then you have some corresponding segmentation. If you can find something overlap, then you can check the cross-section divided by the joint. That's kind of so-called dice score. Of course, the annotation is based on circle, and the protein shape is not necessarily a circle. So you won't be able to get 100%. But the closer you can get, the better the model. So we found if you apply SAM directly, some cases you can actually get very good results. But other cases that you won't be able to see, it doesn't give you any result at all. So on average, you get close to 50%. And then we use the so-called fine-tune model. That's the third generation of AI we talked about. That is that you can fine-tune a large model. Here you have the SAM, and then you just fine-tune the last layer, which is easier, because you can fine-tune everything, but it will be pretty time-consuming. And we found that through the fine-tuning, the overall performance improved a little bit. You can get, some cases, better results. But the problem is that the results shown here, they're really messy. This approach just try to maximize the so-called dice score, but it get so many noises. So that's not what we want. So we designed several prompts in order to achieve better performance. The first prompt is that we use this a dynamic filter just before this foundation model. So the difference between prompt-based learning versus a fine-tuned model that in the case of prompt learning, you do not touch the foundational model. You do not change the architecture. You do not change the weights or parameters in the model. You just add things on top of it. While fine-tune model, you keep the architecture, but you adjust the parameter a little bit. So here we use a dynamic filter as a prompt. That is, we only tune this part. We do not change anything in the SAM model. But when we train the model, we still do it in one shot. This is so-called end-to-end approach in deep learning. So here we use a simple so-called UNet, which is a popular deep learning filter. And then it's actually very simple, only has 2 layers. While many real UNets, they have many more layers. But here, we just want to train a simple one. And then we found through this prompt, even though we do not have a lot of training data, we can significantly improve the performance from 50-something to 70-something. And then if you look at the results, actually, it gives you some really nice results in many cases. But in some other cases, for example, there is actually contamination here. But this approach cannot tell. It actually predicts everything. But this is actually just contamination, not real proteins. A byproduct of this approach is that through this filter, you actually get this prompt image. This image is fed into SAM model. But this image itself has a value because you can see things a lot better. And even for manual labeling, it's better for you to use this image instead of the raw image because you start to see the proteins more clearly. And then another approach we prompt the model is that we use this so-called prefix token. That is in this SAM deep learning model, there are many, many layers of neurons. So we add 1 layer as a prefix of the model. The rest model, as a part of SAM, we do not touch it. And then we just train this prefix prompt that we added. And then we still do the same thing. So this approach actually further improved the model. And then some of the contamination actually can be resolved as well. And another method that we use, in this case, we prompt this encoder. So this encoder has many units. This is called a transformer in deep learning. And then we just add 1 adapter in the transformer. And then we do not touch others. So that means the others, their architecture and the parameters are frozen. And then we just train the adapter. Then we still do the end-to-end approach. So this approach really improved the model even more. You see, this is a ground truth. This is the results, and it's actually getting even closer. So if you look at all these models, you see that the base model, if you don't do anything, you only get like a 50%. And then you do fine-tuning, improve a little bit. And then you use this UNet dynamic filter, improved a lot. And prefix similar, and then the encoder improved a lot. And then you look at the number of parameters. Of course, the last approach is also very expensive because you need to tune every transformer. The number of parameters you need to train is at 52 million. So that requires much bigger RAM in your GPU. Others, you don't need a lot of RAM. Another good thing about this approach is that you actually can do this few-shot learning. So here, we have 470 training samples in total. It's actually not a lot. We could use a fewer and fewer. And then the last approach, even use 5 prompts, 5 training samples, you can still get very good results. So that's actually a big advantage of prompt-based learning. You do not need a lot of data. I mean, even training samples less than 10, you can still get very good results. Okay, so next I will talk about some other foundational models. Nowadays, pretty much any major area, people develop some foundation model. If, for example, it's a protein language model, so people treat protein as a language. we know protein, they are amino acids. You can express them as like this format. And then by training these protein sequences, you can get a protein language model. And then you can use the protein language model to generate new proteins for your protein family or to do other predictions. So here, we actually tried one example. This actually is for signal peptide prediction. This tool actually is very good. It's called Signal P. It's developed by others. That is for a protein sequence. You want to predict where the sequence is cleaved. So this part is a signal peptide. And this is a mature protein. So here, this is the original model they developed. So what we did is that we first of all replace it by a large language model, this ESM developed by Meta. And then we add a prompt. So here, this prompt is like a dynamic filter. But in this case, it's a sequence instead of image. So we cannot use UNet. We use this so-called BioRTM. That's actually a standard way to handle sequence data. So we add this prompt as a dynamic filter, and then we add this prompt into the model. So we found that, yeah, in some small cases, this approach actually did very well. In large category, we have not really outperformed the previous model. And then the good thing that in the model, you could actually pre-train your prompt, this part, by using other categories. So with the pre-training, it actually can further improve this prompt-based performance. So you look at the green, which is actually pre-trained using some big class data. And then the orange is prompt just based on few examples. So you see that after pre-training, the prompt works a lot better. So that actually is pretty much true in all categories. And then the idea that through this approach, we can predict signal peptide in some really small cases. That is, we have less than 10 training samples. We can still develop a model. And then another thing we actually work on that to develop our own foundation model. I mean, developing a foundation model requires massive computing power. So that's why we don't do a lot. But one thing we are actually working on, which actually is published on this archive, is this structure-aware protein language model. So typical protein language model only use protein sequence information. Here, we actually use both sequence and structure information. And the structures are either real structure determined experimentally in PDB or predicted structure in AlphaFold database. And then we train both sequence and structure together and get a new model that reflects both sequence and structure. And the new model actually works really well. For example, in the new model, we can easily separate alpha helix dominant fold and beta sheet dominant fold. While in the language model itself, it cannot separate very well. And then we can even go to the small classes. The point that by using this model to include both sequence and structure, you can do a lot more. We also demonstrate this in several downstream tasks. Another area we worked on is that single cell large models. So there are actually about 10 such large models for single cell RNA-seq data. We actually evaluated them. But unfortunately, we don't think any of these models satisfy foundational model as we talked about. But we still use them for some downstream tasks. For example, in this case, it is so-called SC-BIRD. This model was trained on about 1 million cells. And then we did two similar things that I talked about previously. One is that we do a dynamic adaptation. That is, we put this new data and then some kind of a shallow training, and then put it into the foundation model. Another is that we add adapter on transformers, and we train the adapter. So in both cases, it actually significantly improves the performance. So this task is so-called cell type prediction. That is in RNA-seq data. You have many cells, you have this gene expression, and then you want to predict cell type based on gene expression. So the baseline is so-called single R, which is widely used. And our prompt-based learning that we use some of the labels, and then through this large model, we can improve over single R for cell type classification. So here is a summary and the future work. So I presented two major use cases, one the SAM, one ChatGPT. Both cases offer very useful foundation models for many diverse biological applications. And they are very suitable for hospitals and the labs because it doesn't require a lot of training data. It doesn't require massive computer power. And ChatGPT has a really great capacity as a knowledge graph. That is, you can analyze literary data and study gene relationships or build a whole pathway model. And then we developed a novel approach to automatically optimize prompts, and which actually demonstrate the emerging intelligence of ChatGPT. In a way, this is so-called self-reflection, and ChatGPT has that capacity. And then for SAM, we see in some cases, for example, biological pathway images. So SAM can be applied directly without any modification for image understanding. In other cases, it requires adaptation and better prompts in order to achieve good performance. And in terms of prompts, there can be like either dynamic filter, prefix, decoder, many, as I explained. So for future work, for ChatGPT knowledge discovery, we are using more complex prompts like a tree of thoughts. And then we also are developing a method to use genetic algorithms to fine-tune prompts. So in that case, the prompts may not be even readable to human, but it can achieve better performance. And we are going to study all kinds of molecular relationships, like enzyme-substrate relationships. Then for SAM, we are extending our methods to several other types of images, like the H&E medical images for single cells and the MRI CT images. We actually got some preliminary good results. And then we are also developing this meta learning approach, that is to have some kind of meta prompts for the big class, and then we have individual prompts for individual specific applications. Another big challenge is so-called results grounding or data grounding. That is this model, foundation models and prompts results. They are predictions. And how to evaluate them through existing database, a knowledge graph, or either or even some prediction results to cross-validate. So that's a topic we are evaluating. As I mentioned, the ChatGPT itself as a black box, as an evolving tool, it's actually not very suitable for some of our applications. So that's why we are exploring other large language models, particularly this Llama 2 developed by Meta. This actually is an open source tool, and it can be used for a lot of applications. And we use such a large language models for other type of applications. For example, we are developing some models for RNA, for peptides, etc. Yeah, so this work involves a lot of developers. For image-related work, it's led by Fei He, PhD student at my lab. For ChatGPT, it's led by Yibo Chen, a PhD student jointly supervised by High School and Medical School and me. And then we have a lot of collaborators for the ChatGPT related work, image analysis. Mihail Popescu, Mark Hennig, and Richard Hammer. As I mentioned, Dr. Jianling Chen actually is our collaborator for the core EM image analysis. And we also work with Gangjin Hu at West Virginia University for this programming, this panel GDP, etc. And we got some input from Dr. Mei Chen from Microsoft. This work is supported by NIH. Thank you for your attention.
It's an exciting presentation. Actually, before open a question to the floor, I'd like to raising one of the question, kind of consider the institutional compliance issues in terms of using this is an open source like a ChatGPT. So in case, especially for investigator, so you are studying your own research project with some original data. It's considering with some kind of intelligent property or IP issues belong to the university. If you are doing something like that, so to pump into the ChatGPT, would it be any kind of ownership that's could be adopted by the open source, like open access, open AI? That's the data they can be kind of not using the stolen ways, but so things. If you're loading into the ChatGPT, they will have a memory to whatever the data you're putting on there. So this type of work, how kind of open to faculty or administrator, how that's would be handled?
Yeah, so that's actually a very good point. So for this, I say patient-sensitive data or for proprietary data, that's indeed a concern because OpenAI literally uses all the queries as a part of training.
Yeah,
and then it won't use it explicitly for IP, but implicitly it's included in the model. It actually happened in this Samsung case. That is, employees at Samsung ask ChatGPT some questions that includes proprietary information, but it becomes a part of ChatGPT's training data. And then other people actually can prompt this part. So they can particularly ask Samsung related information. And then people can actually get information accordingly. So that's why I think for this sensitive data, proprietary data, it's probably better to use this open source like Llama or other language model. Even though those models, their performance is still not as good as ChatGPT. But you don't have to worry about such a kind of problem.
Yeah, thank you so much. So,
Dr. Beversdorf online has a comment. It says, related to Dr. Gu's question, NIH strictly forbids reviewers to use ChatGPT to do their reviews in any way.
Yes, that's right. Yeah, I think, ChatGPT. I think it won't be a fair process to use ChatGPT to review papers or proposal because you may overlook some cases. But on the other hand, ChatGPT sometimes can really discover things you may not pay attention to. For example, when we evaluate this fellowship applications, I mean, interestingly, I didn't mean to use it for my writing. I just wanted to test it. It actually gives a pretty good review. Something I didn't pay attention to. For example, one comment given by ChatGPT says the applicant would do research in an institution that is not aligned with his major research interest. And I didn't pay attention to this. Later, I actually studied this institution's research scope versus this applicant's interest. It was not really aligned. So there are some cases they can discover new things, but overall, it's not fair to use it for review purpose.
Thank you. So any questions if you want? Okay, Xinhua.
We trying to use ChatGPT, trying to find topic, for example. Of course, we can collaborate with you. The question is how this is kind of theoretical kind of results from ChatGPT, right? To further direct, the project, for example. how, what do you think how to do this? Say we're trying to, for example, find one gene, like you're saying, interaction, right, with other gene, right? So it's possible we identify the important genes and we can direct, for the future study. So the kind of things.
Yeah, I think ChatGPT itself, as I said, we can build some pathways and relationships, but it's not that strong because it doesn't really utilize a lot of data. So the idea is that if people can use the so-called multi-modality data, and then you can integrate all this biological data, and then in the end, it will have a very strong capacity to make inference for any question you ask. But it's not there yet. I know some people are trying to do that. It will require massive data and computational resources in order to build such a foundational model. I would say maybe in 5 to 10 years, such a model probably will exist.
Okay, thank you. So maybe any other questions? If don't, I have a question regarding kind of your detailed study on the ground truth part.
Yeah.
So you did demonstrate the ChatGPT over the several months, early this year till recently. Actually, the data is kind of frustrating about the output.
Yeah.
So that's indicated. So this is still not quite mature. Even though you're using ChatGPT 3.5, right? So they heard about ChatGPT 4.0 possibly could be improved in some way, but it's still not quite sure. There's some publication recently in Nature about a clinical validation on the ChatGPT 4. However, this is are still debating. So I want to bring these issues because of even though the ChatGPT or a kind of OpenAI type of framework, they constantly acquire data from the community. The major issue is the quality of the data, whether the data is truth or is misunderstanding or misleading. So this part is a big concern at the national level or international community regarding the quality of the data.
Yeah.
So not guarantee all the data put into the public domain are high quality. So this is why early this year, NIH made the mandate for the data curation. So is that the data are be rigorousness, transparent, readable, and interoperable.
Yeah.
So this is a so-called the FAIR principle. So how this type of things or OpenAI, like ChatGPT, would it be integrated because of this is still black box? How that's a part would it be improved?
Yeah, unfortunately, I don't think we can rely on OpenAI or ChatGPT to do the things you talk about. I think ChatGPT or Microsoft, their interest is not really research community because that's not where they can make a lot of money. So they do not tailor research. That's why even though this inference capacity decrease a lot, it's not their focus. They improve other aspects. They reduce the hallucination a lot. So people can chat easily. I think for high quality data or rigorous training, we should really use open source software. Everything will be transparent and will be reproducible. I think that will be very critical. But ChatGPT really opens the door for us to study these things. Once we understand the behavior of the model, things like that, then we should do the same thing using open source software. Okay. Yeah.
Thank you. Okay, question.
Sorry, I know it's beyond your time already. It's 5 p.m., but last question. Sorry. My question is, how do we validate hallucinations compared to the actual reality or real data sets when there's not enough metadata available in the first place?
Yeah, so yeah, that's a good point. So the issue that this hallucination, if you don't have ground truth to validate, then you need to do other methods for the so-called data grounding. This is so-called data grounding issue. There are several approaches. One that you ask ChatGPT about its reasoning. The ChatGPT really tells you how it reached such conclusion. And it also tells you where ChatGPT gets the source of information. And in many cases, actually very useful. I often do that. When I ask questions, I often follow up with the source and then click the link. If the link came from a reputable source, and then tend to believe it better. Then the other way that so-called cross-validation. Cross-validation, you could use the so-called knowledge graph or knowledge base. There are many public databases. For example, in Biomedical, we have tons of databases. So you can actually check with those databases. And those databases typically are ground truth data. So you can see whether there's any conflict. The third way is actually you do multiple predictions. You can use multiple language models to do many predictions, see consensus, or even use ChatGPT by having somewhat different prompts or different setting, you get different results. You see whether there's any consensus. Or you can use a completely independent prediction tool to cross-validate. So by having more tools to cross-validate, typically you can get better results.
Great. Really appreciate it for today's talk. And because of time, let's just give another round of applause to thank Dr. Xu.
Okay, thank you.
Yeah.